So, besides in vitro transcription translation, there was a really interesting uh, uh, paper, and it got Rothman the Nobel Prize, J.E. Rothman, and it got him the Nobel Prize. He was the first one to start going beyond transcription and translation and was actually looking at movement of proteins through the Golgi stacks. And he used it by taking and labeling a sugar that's involved, and we'll talk about the sugar, in acetylglucosamine, which is called abbreviated NAG. So Dr. Bush is nagging you to get to work. Well, there you go. I'm nagging you to learn this, in acetylglucosamine. And what was interesting was they were able, because we already knew there was a virus called uh, vesicular stomatitis virus. It's a cousin of the herpes virus, and it has a protein that has carbohydrates in it. It's called the G protein, glycoprotein. And they could follow, because they had an antibody to it, they could follow this protein through the different Golgi stacks, and they made it in a cell-free system. And so now they had an assay to look at the speed and all the materials required to move a protein through the Golgi. And we'll come back and you can read that. But needless to say, they were able to actually isolate all the Golgi stacks by fractionation. And so now we were making a cell-free system that had basically now a cell-free Golgi apparatus, and we could measure the movement through that. And uh, there are some cells that they use. These are called Chinese Hepster ovary cells, CHO cells. And they had normal CHO cells, and they had a mutant CHO cell. It's called 15B cells. And they were missing a key NAG transferase, and we'll show you how that's all going to work here. And so you probably is going to take you to going through this to come back and read this abstract again a couple times to make sure you kind of got what's going on. I'll let you read this, and then, in class, if we need to, we can go back through this pathway here. But again, this is the first reconstruction of organelle movement and assay in a cell-free system. That's why the Nobel Prize here. So they reconstructed. So this is a reconstruction of transport between the different stacks. So you may not have remembered this from basic biology, but the Golgi looks like flattened stacks, and the stack closest to the ER is said to be the cis. The stack furthest is called the trans, and the one in the middle is called the median. So the medial. So we have cis, medial, and trans. 3 or 4, and they're showing 5 here. And so they found that these stacks can mature from the cis to the trans, and there's also vesicles that can carry materials between these stacks. And we have material leaving the Golgi. This is called post-Golgi cargo. And we have cargo from the ER. So there are vesicles from the ER that are being released and move and fuse into the stack. And then there are vesicles that leave and they have cargo and what. I'm just drawing some cargo here. Then they have some here. And that's called anterior grade. And it turned out, interestingly, some vesicles go back, and those are called retrograde. So now we find that anterior grade moves forward and retrograde moves back. And we're going to get into Golgi structure later, but this is just introducing it in the context of a cell-free system. Okay. So this is a a concept map showing, starting with rat liver. You homogenize by a filter or sieve. You get to homogenate. You use sucrose gradient fractionation, and you're able to purify all the Golgi stacks, a little bit diluted, and you would have to do a second gradient with different densities to get a pure stack. And what we're going to talk about is movement of vesicles to and from the Golgi stacks and within. Okay, that's one of the big red arrow. So that's what Rothman's group did. They were studying here. But once you had pure stacks, that set up how do these stacks stay together and break apart in like mitosis? What are all the proteins involved in the Golgi? That's proteomics here. How does the actin, myosin, filaments, and microtubules, how does all the cytoskeleton work with the Golgi stacks? So those are four big questions, four big cell-free analysis. We're going to focus on the one here, but you can see you could do the other as well. Okay, so I mentioned that and if you review the abstract, you mentioned that we're using a virus protein known as the G protein. It's a G protein because it has a carbohydrate associated with it. Not only is it one carbohydrate, it's a series of stacked carbohydrates, like super thick frosting, if you think about each that. So you may not have remembered the carbohydrate symbols in chemistry, but these little hexagrams all represent a different sugar here. And you can see there is an assembly of this, they call it a sugar tree here, if you doesn't it look like a tree? A sugar tree, and there are special machinery that flips the sugar tree in, and that sugar tree is modified to this final structure. So the tree grows when it's inside the ER. So this is the lumen, this is the cytosol. Here's your protein being made. And at certain points, there are asparagine residues sitting right there. Asparagine is ASN. ASN, that's an amino acid. Not every asparagine gets a sugar tree, but some do. And the formula is an asparagine, any amino acid, a threonine, or a serine. If you see that pattern, then usually if the protein folds correctly, the protein, if it's inside the ER, will get a sugar tree. Okay, and the sugar will be decorated. So there is enzymes. This is an enzyme that takes the sugar tree from this donor structure, which is a fatty acid, phosphorylated fatty acid, and it holds the sugar tree as the substrate. This enzyme takes that sugar tree and attaches it to the protein. And now that protein is said to be a glycoprotein. Some proteins have 9, 10, 11 sugar trees. Some maybe have one. And that's called N-linked carbohydrates, or N-linked glycosylation. N-linked means asparagine, which the one letter code for asparagine is N, just like this is T and S. So you need to start learning your biochem here a little bit. Probably in micro y'all hit this. So it's got a sugar tree on it. And so, of course, they flip the, this is a cytosol now, and this is the lumen. So they flipped it, cytosol, lumen. Now we got cytosol. And inside, so once the protein is folded correctly, then other enzymes actually trim these are 3 glucoses. So the sugar tree is trimmed, right? If you're Christmas tree, you start trimming it, so you put your decorations on. And there's an enzyme called ER glycosidase that removes these 3 red sugars. These are glucose here. And then there's another enzyme called mannose. These purple here are mannose sugars, mannose. And notice it takes these two off. And now all sugar tree containing proteins are moved out in vesicles moving to the Golgi. Okay, so Rothman knew that the VSV-G protein was decorated with the sugar trees. So it's interesting. Here we've got a virus that infects us that actually uses our machinery to decorate its proteins to make them more functionable. So that's not so good, right? It's amazing how the virus has the exact meets the exact requirements to take over host functions, and this is an example. So proteins go into the ER, and that's called glycosylation. They're also known as sugars or carbohydrates. They're inside, and here's what Rothman knew. Not only the sugar tree on the protein, but the sugars are going to be modified in the Golgi. So I already showed the sugar was modified in the ER. Well, the sugar can be modified in the Golgi as well. And they're going to take advantage of that. So that's key. That concept is key for you to understand this cell-free system. And just kind of for your knowledge down the road, most proteins that get translocated inside the ER are glycosylated. Proteins that are made on ribosomes in the cytosol are rarely glycosylated. Okay, the functions of the glycosylation, you're saying, well, why would the sugar be on these proteins? It may help them survive in proteases. If the protein goes to the lysosome, there's a lot of proteases. Sometimes proteins have to handshake with other proteins, and there's some carbohydrate that needs to be involved in that. So those are two reasons. Folding and stability, sometimes moving to the correct department. Lysosomal enzymes actually need a phosphate on a mannose to get to the lysosome. It's the correct zip code. We'll talk about that. And there are diseases where the people are born without the zip code, and it's called a storage disease, and it's not very good. It causes a lot of mental retardation in the kids born because it causes a lot of inflammation, because these proteins that should go to lysosomes are spit out, and that causes all sorts of issues in the body. So some proteins need sugar to become active. Some last longer if they have sugars on them, so the body can regulate the length of time that the proteins are around, right? You don't proteins forever. And it's just showing the asparagine glycosylation. Okay. And this is just showing the oligosaccharide attached, and that's also, so we got sugars, carbohydrates, and it's called an oligo, means many. Saccharide is sugar. Mini sugar tree attached here to the asparagine, that's in residue here. Here's the stacks of the Golgi. And so once a protein is made in the ER, here's the ER, and it's transferred to the Golgi stacks, what happened in the cysts is we start seeing some phosphorylations that can occur. More mannoses are taken off in the medial. Some more, so removal of mannose in the cysts, removal of more mannose in the media. They add a glutanac here, and then they add a galactose and sometimes other sugars, including nana sugar, and then proteins leave going to various destinations. So the trans-Golgi network is where proteins move to different locations in the cell. We'll come back to that. Okay, so that's a key concept. So this is just some of the terminology You'll need this, but this is called the high mannose. Each of the green is a mannose sugar here. And then there are other sugars added with different colors, and just showing how complex some of these sugar trees can get as it moves through the Golgi. So you can have a high mannose, not very complex, to a very complex type of sugar as you're moving. Okay, so flash forward to Rothman's lab. They had been studying VSV-G glycoproteins. The protein is from the virus. The virus comes in, it uses our transcriptional machinery, makes the protein. The protein goes into the ER lumen. The protein has asparagines. It's decorated with sugar trees. Okay, and so in the wild type Chinese hamster ovary cells, CHO. Okay. So this is called the wild type CHO. When they come in, they're trimmed and then they hit the cis Golgi. Here's the G protein. So this is the virus, VSV, vesicular stomatitis virus G protein. Here it is. Some of it's sticking out in the cytosol. The rest is inside. And here's the sugar tree. And so as this moves, notice we start getting a trimming. N-acetylglucosamine transferase adds a NAG residue. That's a red square right there. Adds it. So we're going to add tinsel to the tree there. And then by the time it hits the trans Golgi, we've lost some mannoses. We're down to 3. We've got 3 NAGs, and there's some galactose and NAM in acetylneuramic acid. So this is a very complex sugar tree. This is a very more simple sugar tree. And so this process occurs because there are enzymes in each of these stacks that do this processing. Okay, and again, the VSG protein is put in the lumen of the ER. It has an asparagine that's decorated with the machinery and trimmed, and it moves and gets trimmed, and more sugar is taken away, but much more is added. And then, so this is part 1. Okay, this is the CHO 15B mutants. CHO 15B, I guess that's the strain. I don't know what the other strains were, but this is the CHO 15B cell line you can order. You can order CHO cells, you can order CHO 15B, and that's what Rothman, they had both. And in these cells, something interesting occurs. It shows up with this trimmed sugar tree, but it lacks this enzyme, NAG1. It does not have NAG1. See, right here, key. This is key. Can I say key? Yeah, key. Okay. 15B does not have NAG transferase 1. If it doesn't have NAG transferase 1, in other words, this Golgi doesn't have NAG transferase 1, that means its sugar tree will never have a NAG sugar. Look in the wild type. The sugar's here. It's not there in the mutants. Moves to the trans Golgi. Look what the sugar tree looks like here versus in wild type. Very big difference. If you were to purify, we fractionate Golgi stacks here. We fractionate Golgi stacks there. You immunoprecipitate with an antibody to the G protein and study the sugars will be completely different. They would be, in common, okay, but has 2 more mannoses and will not have any of the NAGs, galactose, and NAM. Big difference. We could tell that difference. Size-wise and other way, we could tell the difference. Okay, so Rothman and them had that, and he says, well, we can fractionate the Golgi stacks from wild type. We can fractionate the Golgi stacks from mutants. And we can then follow the G protein. And so this is their experiment. So this is key, and you can go back and read the abstract. So they took the Golgi from uninfected, that means they didn't add the virus to the Golgi, wild type, CHO cells. And we took infected, VSV-G infected CHO 15 cells and got their Golgi, and we mixed them in a test. So we mixed the different Golgi. Sorry. Getting excited here. So we incubated, and what did they see? They saw that the G protein did get an ag. The only way that would have happened was the protein went to the wild type medial Golgi that they're not showing here. So we got all these stacks here. They're just not showing this cartoon. That means this protein was transported to the wild type, and it got the NAG, and it ended up at the same level here. Don't you love allergies? Okay, so they now knew that there was a transport that they could look from a cis to a medial to a trans. They could now follow that. If the G protein didn't get the NAG, the process wouldn't work. I'm drinking half a bottle here. No, it's water. Sorry. So the G protein is from the Golgi mutant, and it must have gone into the wild type because the wild type had the NAG transferase. And you're saying, what was the incubation? It was cytosol. So they added wild type cytosol here. They're not showing that, but that's the key. And the transport machinery moved the G protein, and stacks between two cell types could function and exchange. You're saying, what happens if the Golgi from the CHO 15 and the CHO didn't interact? Then you would have never seen this, right? It would have gone here, this way, and all the protein would have been in this form. They didn't see that. So they now knew, in the presence of all the proteins in the cytosol, they could measure this pathway here, a cell-free movement of one stack to the next in cytosol. How many proteins and materials are in the cytosol? A lot. And now that's the key to this whole situation that's going to we'll talk about later. But that's the process.